So I'm sorry for any inconvenience that caused. I hope that you're able to, you know, relax or sleep in or whatever, and or go through the practice exam. I think that the practice exam is a very good reflection of what your actual exam is going to be a week from this Monday or a week from today. I'm sorry. Um, what we are going to do now is we are going to be getting into some variations of hemoglobin. So with all of that in mind, let's start talking about hemoglobin, and in particular, um, abnormal hemoglobins. Now, an abnormal hemoglobin, I'm sorry, I'm going to knock my video out just in case I have any internet issues. So I've been having some lately, so I'm just going to kill my video. Um, so abnormal hemoglobins are basically just your variants of your normal hemoglobin, and these are very well known. These are just kind of commonly occurring mutations that you might see. And what I think is most important for you to take away from this is to understand kind of the way that we denote any sort of mutation, and that is by comparing the wild type protein, or the WT, I'll just say, um, to the mutant. And the notation that you'll see is typically going to be, let's say that your wild type protein had a histidine. You'll see the single letter code H, and then the position, let's say that it's amino acid number 17, and it has been mutated to a phenylalanine. You'll see a notation basically just like that. Now, with all that in mind, our hemoglobin mutations may alter hemoglobin's quaternary or tertiary structure. It can destabilize hemoglobin to the extent that the red blood cells cause lysing, causing hemolytic anemia. In addition to that, they can also favor the oxidation of iron to iron 3 plus, so that iron 2 plus Fe2 to Fe3, which is going to cause cyanosis due to the increased concentration of that methylmyoglobin. Now, here's a whole host of maybe the less common or maybe less well known hemoglobin variants. And one of the things that I want you to notice about all of these is they're kind of all named after like locations. And for many of those locations, it's just a matter of, well, this is a mutation that occurred, or it was first identified or first discovered in this location. Um, but I want to go ahead and highlight kind of the effects because whenever personally, the first time I ever heard about it, a hemoglobin mutation, it was sickle cell anemia. And I thought to myself, okay, almost by definition, a hemoglobin mutation is something that's potentially, it's a it's a very significant health issue for someone who's afflicted with it. But here, what we see is kind of a molecular understanding of them. So here, what we have is the Hammersmith mutation, and this mutation is a phenylalanine, position number 42, mutated to a serine. Now, in this class, one thing that I've harped on quite a lot is amino acid structures. And the reason for that is because if we think about phenylalanine, the single letter code for that, or another way of denoting this mutation is F 42S. Now, if we were to classify phenylalanine, we classify that as a non-polar amino acid. Serine, by contrast, we classify it as a polar uncharged residue. Now, if you've ever taken like a genetics class, you've probably talked about mutations and you've probably talked about conservative or non-conservative mutations and basically the implications of a mutation. Um, and here what we have is an example of a mutation and a mutation that would be not a conserved mutation because the chemical makeup of the amino acid phenylalanine, well, phenylalanine as a nonpolar residue compared to a polar uncharged residue. Okay, so phenylalanine is going to love to participate in, hydrophobic interactions, whereas a polar uncharged residue like serine, that's going to more than likely participate in any sort of hydrogen bonding. Now, the implications of this and all of your other mutations vary quite a bit. This Hammersmith mutation weakens heme binding. Okay. So that's not talking about the oxidation state of the iron, but instead the association of that heme prosthetic group with the hemoglobin protein. Now, if we continue looking on at some of these other ones, what we have is let's go with the Milwaukee mutation. The mutation. This promotes methylmyoglobin formation. So I don't know. I mean, whenever I think about this methylmyoglobin, I think about that cyanosis, the blue people of Kentucky in particular, and I'm kind of curious. Like, I've not looked into what the mutation that they had. I'm sure that it's just a Google search away, but um, I'm kind of curious which of these mutations was prominent in that population. Okay, so here's just a variation of or here's just a number of these different mutations. I'm never going to ask you for these specific mutations, like, oh, tell me about the Philly mutation or the Yakima mutation or the Kansas mutation. Instead, I want you to be thinking about this in the context of kind of what we've already talked about, and that is, well, what's changing? What type of what are we going to see occur when we see a non-polar residue switch to a polar residue? What are the kind of what types of interactions will this amino acid now have that it previously did not? Okay. And then in addition to that, think about, well, the basically, you could expect a question from me that's going to ask about which mutation or which amino acid mutation would less likely cause any sort of significant effect. For instance, in this question right here. So here's our first top hat question. The Hammersmith variant of hemoglobin is characterized by weak binding of heme. Okay, so there's kind of our outcome, weak binding of heme. This variant is punctuated by a phenylalanine to a serine mutation. Imagine that phenylalanine, that wild type residue, were mutated to a residue other than serine. Which of the following would still result in a weakened heme binding? For this question, think about what types of interactions phenylalanine and serine are capable of. So basically, this question is asking, well, which is going to cause the same impact as that phenylalanine to serine mutation? So let's go ahead and take a look at that question and try to think of, more or less which amino acid listed here is almost going to give us the same result as serine. So think about the chemical makeup of all these different amino acids as you answer this question. Your 10 seconds left. Right. Okay, everyone. So this, I think, is a really good test question. And it's because it brings back old stuff and it's applying that. So it's kind of applied knowledge of amino acids. Let's go ahead and take a look at the correct answer, then we'll kind of dissect this question. Okay, the correct answer was asparagine. Now, a popular answer was asparagine. Our second most popular answer was leucine. We had tryptophan and glycine. Okay, so let's go ahead and dissect all of these different amino acids and think about kind of their classification. So glycine, I think that if I had basically put glycine or alanine here, that would end up being the most popular or the least popular answer because I think most people, they remember glycine and alanine really well because they're arguably they're the smallest amino acids. In addition to that, these are non-polar residues. Now, this question gave you options and was asking which of these options will still kind of cause that, that weakened heme binding. Okay. So what we're looking for is something that is going to have a similar chemical property as serine. So what we need to know in order to answer this question is say, well, what is serine? Serine is an uncharged and polar. So what that's going to do is that's going to eliminate glycine as a good answer option. Then let's go to our second least popular answer option, tryptophan. Remember, tryptophan's got your two ring structure, and this is definitely a non-polar residue. Tryptophan does have that nitrogen in one of the rings, but still it's a pretty non-polar amino acid because the six-membered ring is your outer of the two rings. Then we've got leucine. Leucine is a hydrocarbon, so that is also a non-polar residue. Now, last but not least is asparagine, and asparagine is a polar and uncharged residue. So, of these four options, asparagine is your best answer option. Now, I'll pose to kind of the chat if I guess if we're looking for, if this were an open-ended question, let's instead change this a little bit and say, okay, any of your 20 proteinogenic amino acids. We're going to make it an open-ended question. Which amino acid will basically provide the result most similar to serine? So you're mutating from phenylalanine to something other than serine, but you want it to be as close to serine as possible. Think about the structure of serine and go ahead and provide an answer. I'll give you a little bit of time for that. So we want it to be polar and uncharged, but think about what serine has. And I'll go ahead and draw serine structure, or it's R group. All label this as BB for backbone. So I'll add a little bit of detail to my question or give a little bit of a hint. What other amino acid or acids, I guess you could say, amino acids have an OH group? Tyrosine would be a good answer option, or threonine. Those are both great answer options. Threonine and THR and TYR. Those are both great answer options because they both have that hydroxyl group. Now, so essentially, if this were an open-ended question, I think that threonine or tyrosine would both be kind of the best options to put down because they both have that OH. They're both going to be polar uncharged. Now, if there was one that I would say is the best answer option, it would probably be threonine. Oh, Morgan, that's a great question. What about cysteine, even though it has a sulfur? Cysteine's also a good option because, if you think about that sulfur is in the um, same column in the periodic table as oxygen. So that cysteine would also be a pretty good answer option. I think that really I would boil it down to cysteine, threonine, and tyrosine. And best answer option, I think I would split it between cysteine and threonine. And I think at the end of the day, if my inclination would be to go towards threonine just because it has that hydroxyl group. And so that's going to be most similar to the hydroxyl group of serine. Now, the only reason that tyrosine, so I would label these as first, most similar, second, and third. The only reason that I would put tyrosine in third place is just because if you remember, tyrosine's kind of got that bulky ring. So that bulky ring and that hydroxyl group, yeah, they're both there, but the bulky ring's going to have, it's going to influence, it's going to fill a different space. So I think this is a very good question because to answer this, you didn't have to have the background knowledge of the Hammersmith variant. All the information relevant to the Hammersmith variant is provided in the question. What you did have to know about for this is the classification of your different amino acids. Now, so this is a good question to sort of anticipate, and it's a good way to kind of test your own knowledge of amino acid structures. Now, we got nearly we got 41% answered correct, so that's good. That's good. But I think that yeah, if you see this sort of question again, don't be terribly surprised. And I like this sort of exercise as a nice thought exercise. Okay, now I gotta clear all the drawings. Now, the more common hemoglobin variant is, or hemoglobin mutant is one known as sickle cell anemia. Now, sickle cell anemia is a mutation characterized by glutamic acid that has been mutated to a valine. So, valine for a glutamate. So, glutamic acid, a glutamate, well, this is a polar, negatively charged residue. Now, if you compare that to valine, well, valine is a nonpolar residue. In addition to the chemical makeup, and this is, these two things are kind of interconnected, but in addition to the properties, so yeah, this is negatively charged, this is polar, this is nonpolar. One thing that's also worth recognizing and thinking about is the similar space that they fill. So basically the size of these different R groups. Valine and glutamic acid are very similar in size to one another. And so they're kind of almost a one for one substitution, um, or a one for one like atom or number of atoms mutation. But then the only difference ultimately boils down to the charge that they carry and the properties that they carry. Now, in the deoxygenated sickled form of hemoglobin, valine, what it's going to do, so this our mutant residue is going to bind in a hydrophobic pocket on another hemoglobin. So here's one hemoglobin, here's a completely different hemoglobin. So these are adjacent proteins. These aren't individual subunits. Instead, this would be HBS. I'll just label this HBS1 and HBS2. So these valines are kind of surface exposed residues. And what do we know about a surface exposed, or what do we know about hydrophobic amino acids? They want to get away from water. They want to find other hydrophobic residues or hydrophobic spaces. And so this is an example of that protein doing as much. Now, this valine is going to find a hydrophobic pocket on another hemoglobin. And this is going to lead to the formation of insoluble filaments that deform erythrocytes, deforming them to the point that they sickle. Now, in oxygenated hemoglobin S, the valine is not exposed, so filaments do not form. So the problem arises whenever you have this deoxygenated form of the protein. Basically, that deoxygenated form of the protein is at a disadvantage. If you think about deoxyhemoglobin from the get-go, it has a really low affinity to oxygen. It doesn't really want to bind oxygen. And then if you make it, or if it ends up like uh, forming these filaments and crystallizing with other proteins, then it's really up creek. It's really in trouble. Now, the deoxyhemoglobin S filaments dissolve upon oxygenation, so no fibers are present in arterial blood. But over time, these um, filaments become stronger and stronger and larger, and they form fibers, which ultimately can lead to pretty catastrophic effects. Now, I'm going to clear all this out. What type of interaction is established between valine and a phenylalanine? We got about 15 seconds left. Hey, where did we go? Where did we go? Where did we go? Are correct. Okay, hydrophobic. Great job. Okay, so this is a just kind of another example of can you classify your amino acids? And you know, I would say that that is in terms of importance of understanding biochemistry, being able to classify and identify your proteinogenic amino acids is maybe one of the most, it's on par with being able to balance the chemical reaction or understanding the concept of a mole in a gen chem class. It's like you can, this is going to kind of, uh, it's going to persist and it's going to basically follow you through the entire class. Okay, so hydrophobic is the right answer. These R groups are both they're both nonpolar residues, so it's not going to be a covalent, ionic, or hydrogen bonding. All right. Now, sickle cell anemia and the sickling of cells, some hemoglobin S homozygotes have or show only a mild form of sickle cell anemia, and they express high levels of fetal hemoglobin. Now, one thing, if we think back to, I guess it was last Wednesday, HbF, or fetal hemoglobin, what that does is that's a form of hemoglobin that has, well, one thing that it has is a low EPG, bisphosphoglycerate affinity. And so, in effect, this actually has a high affinity to O2. Because if we remember, one of the things that we mentioned last week was fetal hemoglobin and I guess the maternal hemoglobin, they don't really fight for O2. They have similar affinities for O2, but by virtue of HbF, that fetal hemoglobin having a low affinity for bisphosphoglycerate, in effect, HbF has a higher affinity for O2 than the maternal hemoglobin. Now, one thing, if if someone is afflicted with and they are homozygote for hemoglobin S, um, then they're going to express higher levels of fetal hemoglobin. And so that can kind of compensate for their deficiency in their hemoglobin's function. So, one thing that can be done is this compound right here, hydroxyurea, is actually something that induces the production of fetal hemoglobin. So, when that's administered, that can induce expression of fetal hemoglobin, and so that can be used almost as a therapy or an intervention. Now, malaria is something that if someone is afflicted with hemoglobin and they are a hemoglobin S heterozygote, they're more resistant to malaria than uh, people with wild type hemoglobin. Malaria is caused by a mosquito-borne protozoan, which resides in erythrocytes during its 48-hour life cycle. Now, infected erythrocytes adhere to capillary walls, causing death by impeding blood flow to organs. The protozoan causes a decrease of pH by zero point 0.4 units. Now, if you remember when we were talking about the Bohr effect, with the Bohr effect, one of the things that we noticed was that I think that the range of pHs was basically like 7.6 down to 6.8. The Bohr effect is kind of punctuated or characterized by a drop in pH causes a drop in O2 affinity. Now, another way to put that is it causes P50 go up. So essentially here what we have is a protozoan that causes a decrease of pH effectively enhancing the Bohr effect. The lower pH is going to favor that deoxyhemoglobin or your T state of hemoglobin, and the parasite-induced sickling allows the spleen to remove the infected erythrocytes from the system. So this is something that someone who's a heterozygote for hemoglobin S is capable of basically fending off. All right, now we're moving on to muscle fibers. Okay, so I'm going to take a drink of my coffee real quick. So, muscle fibers. Now, muscles are composed of bundles of large or singular large cells. Now, the major parts of these cells are depicted here. There's a single nuclei, there's striations, sarcolemma, sarcoplasm, myofibrils, and myofilaments. What we're going to be looking at is the movement and the contraction and relaxation of these. Muscle fibers are formed by cell fusions and contain, multiple nuclei, not single nuclei. I apologize for that. Each fiber contains many myofibrils. So these muscle fibers have a large number of those. They are composed of repeating units called sarcomeres. So think about the singular unit is the sarcomere. And these are bundles of actin and myosin filaments. Now, everything that we're going to look at, or for the most part, what we're going to be looking at with these cross sections are basically combinations or not combinations of actin and myosin. Now, these are what we've mostly been talking about is from a protein structure standpoint. We've been talking about globular proteins. Here, what we're going to do is we're going to pivot and we're going to be talking about filamentous proteins. Now, one of the big differences between a globular protein and a filamentous protein is that your globular proteins, they're kind of just that. They have kind of an amorphous shape. And there's a little bit more, I guess you could call it disorder, more disorder. Your filamentous proteins, on the other hand, they are more rigid in their structure, and they are, well, they're more rigid in their structure, and more, I guess, well, let me move where what I'm going to write there. Um, they have more of a pattern. I'm going to say have a simpler pattern. And to kind of characterize that with our globular proteins, when we looked at hemoglobin, HB, and myoglobin, MB, we had that shape that we referred to as a globin fold. Okay, so with a globin fold, that's basically, yeah, you have lots of alpha helices. Okay, and maybe there's 9 or 10 alpha helices. The difference between a globular protein with a series of alpha helices interrupted by, I'm going to say, alpha helix and random coils. With a globular protein, you have alpha helices and random coils. Sure, filamentous protein, it's going to be basically one alpha helix. And I'm going to say that's one alpha helix, and then I'm going to say that's on into eternity. As soon as that alpha helix ends, that protein's structure ends. So that's one of the biggest differences. In addition to that, what that leads to is something that's more rigid in structure. So your filamentous, or also known as your fibrous proteins, those are proteins that are going to have they're going to give like structural integrity to a cell or structural integrity to something like a singular muscle fiber. Now, our myofibrils are muscle fiber components. I'm going to clear all my writing off the side here. As is always the case, this is recorded and I will post it on our Canvas site a little bit later today, at least by 10 p.m. tonight. Try to do it before then, but there we go. Now, if you look over here on the right, this is a depiction of some of these repeating singular units known as sarcomeres. So here what we have is a single sarcomere. Now, one of the things that's kind of challenging is that this from here to here, this is part of a muscle fiber. I'll just say a fiber. And this actually extends on this end and it extends on this end. But this is just sarcomere after sarcomere after sarcomere after sarcomere. So we have lots and lots of sarcomeres. Those are our repeating units. Now, a sarcomere, it has boundaries. And the boundaries of a sarcomere is what is known as a Z disc. I always kind of think about those as fences of sorts. Now, within a Z disc, you have three major components. And I'm going to dissect each one of these in just a little while because each one of these is basically um, a composition, is composed of actin and myosin filaments and, where do they overlap or not. Okay. Now, the the bookends or the fences of a sarcomere are the Z discs, and the midpoint of a sarcomere is known as an M disc. The innermost region of a sarcomere is the H zone. The kind of flanking sides of a sarcomere are the I bands, and the interior region that is composed of the H zone and then some is known as the A band. Okay, so these I think are helpful to kind of basically work your way out. So I always think about it as M disc is in the middle. The H zone overlaps the M disc. The A band overlaps the H zone and the M disc. Your I bands are on the periphery, and those overlap your Z discs. Okay. Now, what this means and what all these different things are is shown here. They're composed primarily of the proteins actin and myosin. The I bands, so your adjacent regions, what these contain are only very thin actin filaments. Okay. The A bands also contain thick myosin filaments. So when you think about basically what this is composed of, is an A band is actin and myosin. The I band is actin only. The myosin and actin filaments overlap in peripheral regions of the A band. Okay, so if we take this depiction, this region right here, myosin and actin filaments are going to overlap in the peripheral regions of the A band. So this that I've just circled, that is actin and myosin. So this A band covers from here all the way to here. Within this region right here and this region right here, it's actin and myosin. Okay, now I'm going to go ahead and write right here. This is actin, and then this is AM, actin. Now, what that leaves is, what about the H zone? The H zone is myosin only. So basically, the A band is what links the you could think about it almost as the A band links the I band and the H zone to one another because that's where you have these overlapping regions. Now, what's significant about this, like, is this is how muscles contract and expand. They contract and relax. The actin filaments are attached at their plus ends to the Z discs. So actin links right here, here, here. These are their plus ends. And the same thing on the other side of the other Z disc. The myosin filaments are anchored at the M disc or M line in the middle of the sarcomere. Now, a, a good question, a fair question to ask is, well, how am I going to be asked about this? How am I going to be kind of evaluated on this? Well, one way is by being able to identify what are these proteins here and these proteins here. Well, being able to see that this is within the I band, then it's safe to say, okay, well, the I band only has actin. So this singular filament right here, and all of these ones below it, those are actin. Now, what about within this region, the H zone? Those are only myosin. So these thicker bands, those represent myosin. So that M disc is the middle, or I always kind of think about it as myosin anchoring band. That's where that protein is going to be found. Now, all of this is helpful and all of this is good to know because what's happening right here is that your muscles are going to contract and relax based on a interaction between actin and myosin. Now, what I'm going to do right now, so I think I'm just going to, yeah, okay. So we'll do a question, then we'll talk about Titan and Nebula. So let me clear this out real quick and jog back for a second. Okay. So what's going to happen is that this right here, this interaction between actin and myosin, it's going to release, or this interaction gets disrupted. And then what happens is that basically these proteins creep towards one another. And one of the ways that you could think about that is the Z discs in muscle contraction. Z discs get closer to one another. These Z discs are going to get closer to one another, and that is going to result in the Z discs are, sorry, the Z discs are going to get closer to one another because where actin and myosin overlap with one another, there's going to be a release. And then think about it almost like myosin is going to drive this way. It's going to drive to the left. And then over here, this myosin is going to drive to the right. So I'm going to turn my video on here. Imagine that it is, and I'm going to do my best visual of this as I think that I possibly can, but it's almost like my right hand here is myosin. What it's going to do, and here's a poster. So my hand is myosin. It's going to release. And then this actin filament is going to shift this way. And it's going to attach again. Then it's going to release, shift, attach, shift, or sorry, release, shift, attach, release, shift, attach. So that's how a muscle is going to contract and relax. Now, what you're going to, and we're going to skip that last question, and instead I'm just going to kind of explain it. So if we have each of these regions, if we have actin and myosin kind of contracting and expanding, what do you think is going to happen to, let's say, the H zone? Will the H zone get larger or smaller? And add to the chat what you think that is. You're absolutely right. The H zone is going to get smaller. Okay. Now, what do you think this means for the eye bands? Are they going to get larger or smaller when muscles contract? Okay. So the eye bands, keep in mind that actin and myosin, they're still kind of going in opposite directions of one another. So this guy right here is going this way, and then this right here is going this way. So the H zone's getting smaller. So too is the I band. The I band's getting smaller because if you think the overall, let's think about, and I think that this is really helpful, are the Z discs getting larger, or sorry, getting closer or farther? They're getting closer. So if the H zone is shrinking, so too is the I band. Now, if we think about, I'm going to clear my drawings out here. I'm going to give you some options. We've got the I band, H zone, and the A band. So we talked about the H zone. The H zone is shrinking. The I band is shrinking. What's happening to the A band? Morgan, absolutely right. Darlene, absolutely right. It's getting larger because the space, like this region right here, well, the overlapping region between actin and myosin, that's growing. And so what that means is our A band as a whole is getting larger. Okay. Well, I think that's a good place to stop. We're gonna do some more dissection of muscle fibers on Wednesday. Um, since we didn't have class on Friday, again, I apologize for that. I never like to do that. But I don't want anyone to like we're still kind of on track with where I want to go with the for the exam. We might do a little bit of a lecture and then review on Friday as opposed to just review. But I think we're in a good place still. All right, everyone. I'm going to go ahead and stop my recording. I'm going to stop my share. I hope you have a wonderful day. Thank you so much for coming to class. And if you get a chance, go to the football game tomorrow night. Or don't, just enjoy it. Just enjoy the day. Thank you so much, everybody. Bye.